And then one of the days when I started teaching here, I wanted to wear a bow tie. And I know actually how to tie a bow tie, but it's difficult. I refuse to wear clip-ons. So, and I went to the Kentucky Derby a couple years ago, and I got up 2 hours early, and it took me close to an hour to tie my bow tie. And I was determined to do it. All right, so let's talk about tissues. This is chapter 5, in case you were wondering. What I'm going to do is I'm going to go over some of this material, kind of like a pre-lab material, all right? And then, as I see your eyes start to glaze over and your brain start to get full, we'll go into labeling. That usually works best for you guys, for the students. When you're kind of alert in the beginning, we'll take the notes and I'll teach. I'm always teaching, and then we'll transition over into the labeling portion. All right, so let's talk about tissues here, okay? So a couple things you want to know about tissues is when we're looking at tissues, one, we're going to see a cluster or grouping of cells that are similar. And then we're going to talk about the material that's outside the cell. So we refer to that as extracellular material. Now, extracellular material is broken down into what we refer to as protein fibers. We'll talk more about this in a little bit when we get into connective tissue. Protein fibers, all right. Oh, it's hard writing with a mouse. All right. And then the other part that makes up the extracellular material is called ground substance. Okay. I'm not going to write the whole thing out. Ground substance. So protein fibers and ground substance make up the extracellular material. And then extra meaning outside. So that's all the stuff outside the cell. So we can't forget the cells themselves. So anyways, these tissues will perform a common function: secretion, absorption, filtration, all right? An example here is protection, okay? So they'll have some sort of common function, all right? So we mentioned this the first week of class when we were studying tissues, that it's called histology. Now you need to know this. This has been asked on tests many, many times. There are 4 types of tissues, and if you're unlucky enough to have me for 211, I will pound this into you question after question after question. All the time, I'm always asking my students, what are the 4 tissue types? You need to know that. Epithelial, which we're going to talk about today, connective, and then the last 2 are the easier ones, muscle and nervous tissue, okay? Those are the 4 types of tissue. So they look different, all right, and they act differently. So they have varied structure and function. All right? So the first one we're going to talk about is going to be epithelial tissue. And I'm going to teach you how to name it. Okay, we call it the nomenclature. And there's 2 components to the naming of epithelial tissue. Okay? So the first one here is the number of cell layers. Okay, that's the first part. There's a second part. So we're going to talk about the number of cell layers when we're talking about epithelial tissue. And we'll get into the characteristics in more in detail on the functions in lecture class. Okay? So if it's only 1 cell layer thick, all right, it's simple. Simple will be in its name. Okay? 1 cell layer thick. And epithelial tissue, I'm going to come right back to the slide, don't freak out. Epithelial tissue sits on a basement membrane. That's this kind of pinkish structure here that you're seeing. So here you can see the cell layer. It's 1 cell layer thick, and it sits on this basement membrane. That's one of the characteristics of epithelial tissue. Okay? So when it's simple, 1 cell layer thick. All right. So when we're dealing with simple epithelium, primarily, all right, their functions for simple epithelium are going to be filtration, absorption, or secretion. All right. Filtration and absorption are pretty straightforward. You'd much rather have one cell layer of thickness if you want to filter something in the body or absorb something in the body. You think it's easier to move a molecule of oxygen through one layer of cells or 50 layers of cells? What do you think? One, right? Yeah. Is it easier to climb up one stair or 50 stairs? That's right. Exactly. Thank you at home. You're absolutely correct. So same thing with absorption. Okay. So simple epithelium is best for filtration, absorption, and secretion. It wouldn't be very good for protection, right? I mean, if your skin was one cell layer thick, it would be bursting apart all over the place. All right. Thank God it's not. Okay. So the other name that we give to epithelium is stratified, and that's if it's 2 or more cell layers thick. Yes, sir.
So, like older people, is that why they like bruise and like their skin?
That's a great question. And it's funny, not enough funny. There's nothing funny about that. But when we get into chapter 6, we'll discuss that. But it has to do with connective tissue, which is the second type of tissue that's in the body. You start to get less amounts of connective tissue. You've heard of collagen. It's a protein fiber. It's a very strong protein fiber. It's like a cable thickness. Your body starts to produce less of that. And that's what helps give skin its resiliency. It's, you know, and so when you have poor quality of anything as we get older, it's unfortunate, but our bodies just don't make stuff as well as they used to. And so you talk about the bruising, yes, blood vessels become much more fragile. They're not as flexible, so they're more rigid. And so they can't handle the stresses that they used to, so they can become damaged. And the same with the skin, same type of thing. You know, the skin becomes thicker because you're producing less epithelial cells for the skin and your connective tissue is less. So yeah, great question. Okay, so with stratified epithelium, there's so many things I want to say right now. For stratified epithelium, the basal layer, that means bottom layer. Okay, basal is bottom, apical is the top. Okay, is going to be in contact with the basement membrane. So I'm going to come back here. Here we can see in our picture here, here's our stratified epithelium. So here is the bottom layer right here. It's in the book, it says it's the basal surface, but it's the basal layer. And then up here at the top is the apical layer, or the apical surface here. Right? So the apex of the mountain is the top. That's how I remember it. And the bottom of the stratified epithelium is the basal. Both begin with B, BB. Okay? So if you look here, and I'll say this now because I might forget it later. If you look here, as we zoom in here, and this is for you guys at home too, look at the shape of these cells here versus the shape of these cells here. Are they the same or are they different? Different. Good. All right, we can all agree on that. All right, they're different. So, knowing that, when we're talking about stratified epithelium, we always name it. We haven't gotten to this part of the nomenclature, all right, but we always name it for the appearance of the apical surface here. See right here? Okay, so these cells look flattened and these look more kind of square. And we'll talk about the classification part in a second. All right, but I just want to say that now in case I forget. I don't want to, that's very important. So the apical surface, or the apical cell layer here, that's what we name the tissue for its shape. All right, we don't talk about it down here. All right, and you'll see why in a second. All right. Here. Oops, one more. Okay, so we have stratified epithelium, 2 or more cell layers. The bottommost layer is in contact with the basement membrane. All right, and then this type of epithelium is great for mechanical stress. Remember, it's multi-layered. Your skin, like I said, one layer versus multiple layers for the skin. You much rather have multiple layers because it can handle that mechanical stress. One of the characteristics of epithelial tissue is it has polarity. Now, I'm not talking about a negative or positive charge, okay? Just like the Earth, how it has a North Pole and a South Pole, all right, they're just different parts of the same structure, all right, the Earth. Well, for a cell, you have an apical surface, which is the top surface, all right, and then you've got the basilar surface, or the basilar part of the cell, which is the bottom portion. You want to know that. So you'll hear me say terms like columnar epithelium. Those are long, skinny cells. The nucleus will be at the basilar portion of the cell. And all I'm saying is at the bottom portion of the cell. It's very unique to epithelial tissue. Yes, it's one of the characteristics, and we'll talk about it in lecture. But I just wanted to talk about it now in case I forget. All right. And then the last type of epithelial tissue, for classification based on number is this guy, pseudostratified. Now, pseudo means false, fake, okay? So it's falsely stratified. It appears to be layered, but it's not, okay? But here's the thing that you need to know, and I've seen this asked in a test question. How do you tell the difference between stratified and pseudostratified? Here's how, okay? In stratified epithelium, the only cells that make contact with the basement membrane is the bottom layer, okay? In pseudostratified cells, all right, it appears to be layered, but all the cells will connect to the basement membrane, but not all of them will reach the apical surface, okay? So it looks to be layered, okay, but all those cells are connected to the basement membrane, but they all don't travel to the apical surface. Now, in this picture here, all right, you can see that you have this layer of cells down here. They're all connected to the basement membrane, but eventually all these cells will make it up to the apical surface, okay? But, well, that's for skin. Strike that out. Don't write that down. I'm getting too far ahead of myself. This is what you need to know just purely for when we're talking about pseudostratified, all right? Not all of them will make it to the apical surface, all right? And that's the reason why they look very similar and they're hard to differentiate, okay? So if you were to be asked on a test to identify, it'll be obvious, all right, that you can tell that certain cells will not touch the basement membrane. In stratified epithelium, okay, not all the cells touch the basement membrane, okay? If the apical layers, the top layers, won't touch the basement membrane, okay? But in pseudostratified epithelium, they all have to touch the basement membrane, all right? So to differentiate, kind of go back to who asked me that question. Alexa, is it, well, I can't hear what you're saying. Is it Alexia or Alexa? Watch her just type out her name again. Oh, it's Alexa. Is it the first one or the second one? Alexa? Alexia. I think that's right? Did I pronounce that right? Yes. All right, Alexia. Sweet. All right, I'm Polish. I got it. All right. So keep in mind what I just said. All right. Stratified epithelium. Okay. Let me make sure I got this right. Stratified epithelium. The apical cells will not touch the basement membrane. Okay. But in pseudostratified, all the cells will contact the basement membrane. Okay. All right. The second part of classifying these cells, the first part was by the cell number. This one is by the shape. Okay. So the first shape that we're going to talk about is squamous. Squamous cell kind of looks like a fried egg. You'll hear people refer to it as a tiled appearance. So it'll look flat. Okay. And the nucleus will look flat. So I'd say it looks like a fried egg. The next type is called cuboidal. So it's going to look like a cube. It's going to be as tall as it is wide. The nucleus will usually sit right in the middle, and it'll have a spheroid or spherical shape to it, like a golf ball or a baseball or a softball. Okay? Columnar epithelium is like a column, taller than it is wide, The nucleus likes to hang down in the bottom half of the basilar region, and its nucleus looks like an egg, kind of oval in shape. Okay? So far, those are pretty distinct differences, right? You'd say. Okay. The fourth is the transitional cells. This is the transformer epithelium. All right. These cells will literally change their shape, and I'll get into more detail about it in the beginning, not in the beginning, later on. All right. But it depends on what's going on. You find this type of epithelium in the urinary bladder. So if your bladder's full, okay, then they appear flat. All right. When your bladder's empty, they're going to appear more rounded. And I'll explain to you a little bit more detail when we get to the transitional. But just know, all right, its shape depends on what's going on to the epithelium. Okay? What kind of stress? If it's a full bladder, they'll be smushed down. If it's an empty bladder, they'll be more rounded and they won't be compressed. But it's the first three, these guys, that I really want you to be concerned with. You want to know those. All right. Let's talk about, here I showed you, all right, stratified epithelium, one cell layer thick, all the cells make contact with the basement membrane. Here's our stratified epithelium, all right, many layers, 2 or more layers, all right, only the basilar layer, the bottom layer here is going to make contact with the basement membrane, all right, but then again, we always name, all right, epithelium after the appearance of what the top layer looks like. So here now we know these look more like they're cuboidal, all right, at the basilar region. But when we get up to the apical region, they look more squamous. So therefore, we would name this stratified squamous epithelium, okay? Because these cells have a squamous appearance. These guys down here have more of a cuboidal appearance. Here's the 3 cell shapes. Here's the squamous cell. Here's the cuboidal cell. Here's the columnar cell. Okay? So now we're going to start combining these terms. So we're going to learn about simple squamous epithelium. All right? Stratified cuboidal epithelium. So now we're going to start combining these. Now, let me point out, this is figure 5.3. You definitely, epithelium is very intimidating at first glance, because if you look, and when we start to go through this, some of you might get overwhelmed, and that's normal, and that's okay. So what I would say is, look at this chart here, look at the different types of epithelium, okay, just to kind of get an understanding of epithelium, all right, and then you can start to learn more of the details. So I would say, if you're into the memorization thing, memorize this chart, okay? When you have epithelium, start off with simple and stratified, and then you can look down at the subcategories, like simple squamous, simple cuboidal, all right? And then you can just learn about a few of the characteristics here, all right? And then you can move into more of the specifics here. Just start with this so you're not overwhelmed. We're going to start to go through, today we're going to go through all the epithelium for lab. So we're going to start to go through all these different specific types of epithelium. Okay, and hopefully it won't be overwhelming. All right, I always hope. All right, so let's start off with simple squamous epithelium. Okay, so you look at the name. Simple, how many cell layers? Without reading the slide. Right, and what's the shape of the cell? Flat. Okay, so the nucleus will be flat. The cell will be flat. The nucleus will be flat. It'll be, yeah, like an egg, a fried egg. All right. So this type of epithelium is going to be found, it's the thinnest type of barrier that we can have in our body. All right? In fact, we find it in the alveoli, the air sacs of your lungs. Because remember what we said, simple epithelium is great for filtration, absorption, and secretion. So what transpires in your lungs? What occurs there? Gas exchange. That's right. Your respiratory gases get exchanged between oxygen and carbon dioxide. It flows across. So again, remember what I said. It's much easier for these respiratory gases, and I'll zoom in here, all right, to move across this tiny thin barrier here. Okay, this is the alveoli. These spaces here, this is where your air is coming into. All right, there's a simple squamous epithelial cell. Over here, you can see off to the side, this is a capillary. Okay, so your blood will be traveling through, all right, here, all right, and it'll cross over one layer of tissue, which is nice. You want Your body always wants things to be as easy as possible. Nature is like that. It's lazy. It really is. So you want to expend as little energy as possible, and you want to make things as easy as possible, all right? So normal breathing is almost effortless. I mean, essentially, when you're taking a breath in, the only muscle, well, there's a few, but the major muscle is the diaphragm, and it contracts. And when it does that, it creates less pressure inside your chest than the pressure in this room. So we're going to learn this relationship in chapter 4, but things like to go from an area of high pressure to low pressure, or an area of high concentration to low concentration. So when you contract your diaphragm, it makes the pressure inside your chest less, so air flows into your chest from high pressure to low pressure. And that's what happens. You'll learn it more in detail when you do respiration. But that's what we're seeing here. So it's nice and easy for gas to cross over that barrier there. All right, so this is what we're talking about. You're going to find this type of epithelium, all right, in your lungs, in your vessel walls, and it doesn't say blood vessel walls, vessel walls. That includes blood vessels and lymph vessels, okay? We'll learn about lymph system in 211, but we refer to this type of epithelium as endothelium. You need to know that, okay? So the lining of your of your vessel walls, that's lymph and blood, is referred to as endothelium, and that is simple squamous epithelium. We learned about this last class. Remember serous membranes, right? Those membranes that line the body cavities. Okay, we talked about the parietal layer and the visceral layer. All right. Well, anyways, we refer to that type of epithelium as mesothelium. You ever heard of mesothelioma? It's cancer of this type of epithelium. Right? You hear it mostly in relation to the lungs, right? But that has to do with mesothelium has to do with the serous membrane. Okay? Yes.
What is the basement membrane?
The basement membrane, I don't want to call it like a connective tissue, but it's connective tissue-like, or connective tissue-esque. It just sits at the right on the basilar portion of the epithelium. And then what you can have right below that is connective tissue, all right, specifically, which we'll go into in the next, well, we won't do it today. You can have loose connective tissue, dense connective tissue, but it's just this layer that of tissue. It separates, and there's 3 layers to it. I won't get into the specifics of that. Well, I might talk a little bit about it in the lecture if we have time, but it's always just right beneath the epithelium there. So these pictures, by the way, just to let you guys at home know too, these pictures are in your textbook. They're a great reference because here we go. You can see this gives a little bit more of a description. I know I have some of the descriptions up here, but this gives a little bit more of the description for the function, where you'll find it, okay? All right, so the next type of epithelium is simple cuboidal. So again, when we look at it, you see the names, simple, you know it's one layer. Cuboidal, you know the shape of the cells, all right? So I can kind of skip that right here, all right? One layer of cells, all right, they're as tall as they are wide, and the nucleus is centrally located, a nice spherical shape. And if you look at these examples here, these are beautiful cuboidal cells here. You can see nice square-shaped structure with a nice spherical nucleus here. So this type of epithelium is great for absorption. You see it in the kidneys, right? Because we're going to get a lot of absorption occurring in the kidneys, and secretion, because when we absorb something, we can sometimes are going to exchange it with other things. Our glands in our body, sweat glands, some of our endocrine glands, are made up of simple cuboidal epithelium. And so it's great for secretion of whatever we're making. If it's going to be sweat, it's going to be a hormone, all right? So we'll see these in high amounts or decent amounts in glands, okay? All right, and then we got simple columnar. Now, remember, columnar is going to be a tall cell, so it's going to look like this, okay, like a column. And you see the nucleus will be at the bottom there, or near the bottom. So again, it's simple, so it's one layer, all right, taller than it is wide. The nucleus will be what we refer to in the basilar region, and it'll look like an egg. So this type of epithelium, again, is going to have a role of secretion and absorption. So there's two types, or two forms. We've got our non-ciliated and ciliated. Does anyone know what cilia is? Yeah, it's like a hair-like, yeah, exactly. So there's three what we call apical modifications, which is the top part of the cell. And you can have three types of apical modifications. You can have what's known as a microvilli or brush border. You can have a ciliated modification. It's going to have these hair-like structures. And then you can have a flagella, which is one big, long process that comes off. Okay? So we're going to talk about the first two. Okay? So this would be a ciliated form of simple columnar. You have these long hair-like structures. All right? And this would be a non-ciliated. All right, so we'll talk about those. Let's start off here with the non-ciliated. So this means it does not have cilia. It does not have these long hair-like projections coming off here. Okay? So this type of epithelium has a structure on top which is called a microvilli. All right. We also refer to it when we're looking at it on a microscope as a brush border because it has this fuzzy appearance to it, all right, like a paintbrush. All right. So one of the things that we associate with this type of epithelium is a specific gland, all right, which is called the goblet cell. So have you ever seen a goblet glass? It looks something like this. Almost like a wine glass. And it has just a slender portion to it down here. Okay. And the nucleus of a goblet cell will sit at the bottom, kind of like this. I'll show you. Okay, it's a horrible picture. But anyways, there's a glycoprotein in it. Oh, what's a glycoprotein? Simple. All right, it's a protein with, look at the first 5 letters, sugar, carbohydrate. Okay, so it's a protein with a carbohydrate attached to it. And this specific glycoprotein is called mucin. And it lives here. Okay? So, what happens is this cell will push the mucin out, all right, into the lumen or whatever, and it'll mix with water and it'll become mucus. So, don't be fooled. Goblet cells do not produce mucus. They make mucin, and then when it mixes with water, it becomes mucus. Okay? Just read the question, all right? If you're asked that on a test, and if you have 2 answer choices, and one is mucus and one is mucin, all right, they make the mucin. Okay? All right. So mucus, and you've learned, all right, today that mucus does, well, you learned this one part of it. Mucus will act as a lubricant, but it also helps to help neutralize some of your stomach acid. Okay? So, point being, let me just erase this. All right. So, if you were to take, and I'm used to drawing all this stuff on the board, so bear with me, I'm using the mouse here. Here's a columnar cell, another columnar cell. If we were to measure the distance from this part of the cell to this part of the cell, just the apical surface, let's just give it an arbitrary number. It's 10 micrometers. It's very small. Okay? All right? And say that this cell's job, all right, is to absorb stuff. So I only have 10 micrometers to absorb stuff. So I'm limited. So what if there were a way that I could actually increase that surface area so I can absorb more stuff? Your body did it. Okay? It took my idea. And what it did is it created this, which is the microvilli, which is this brush border. All right? Now, this is a horrible version of it, all right? But that's supposed to be all uniform. Okay? So imagine I took a string, all right, a really tiny string, and I stretched it this distance here. And it measured 10 micrometers because that's the distance from here to here. Right? People at home can't see me doing this. That's the distance from here to here. Okay, so here's my piece of string, 10 micrometers here from here. Now, I'm going to take another piece of string, and I'm going to start it here, and I'm just going to follow the outline of the brush border. Whee! And I do that, and I do that, and I do that. All right, and then I cut the string. And now I'm going to take that string, and I'm going to stretch it out. And it measures out to be 15 micrometers. So your cell has quite cleverly figured out a way to, even though it has that 10 meters here of real estate exposed to the lumen, it's managed to now take advantage of that and create this brush border and increase its surface area for absorption. Clever. That's what the rugae are in your stomach. Their function is to increase the surface area for absorption, all right, in that small confined space. Well, that's what the microvilli have done here, okay? All right, so we're going to see this type of epithelium pretty much from the bottom, from your small intestine, all right, from the stomach down to your anus. Yes, ma'am.
Is the left square, is that non-ciliated?
Are you talking about here?
No.
Oh, no. Yeah, this was an example of non-ciliated without a microvilli, without the brush border. Yep, all I'm just showing you is how the brush border, and there are non-ciliated that doesn't have a microvilli. All right, but I'm just showing you the difference between the two. All right, so let me zoom in here real fast, kind of show you a few things. So this, again, this white area here, this is the lumen of your small intestine, okay? So you can see, here's a columnar epithelial cell, all right? Look at the nucleus is at the bottom of the cell, okay? And you can kind of tell right here, that's your brush border, right there, right there, right there, right there. Okay, that kind of pinkish, fuzzy area there. And then when you see areas of this whiteness here, all right, that's a goblet cell, and it's filled with mucin. And when that mucin gets ejected out here into the lumen, all right, the small intestine, it mixes with water and it turns to mucus. The lumen is the space. Take a toilet paper roll, that space in the center of the toilet paper roll, that's the lumen, all right? All right, questions? Cool. Yes, sir.
I'm still confused, like when you said like the cell, like it can reach out 5 more micrometers instead of 10, so like it kind of stretches itself out without actually moving it.
Right. So if you see, all right, here, using this string, if we trace from this side of the cell to this side of the cell on this cell, all right, and we use like a string just to trace it, and we measure that, it comes out to like 10 micrometers. It's not a specific number. I'm making that number up in my head, all right? Then we look at a microvilli cell, all right, that has this brush border, and it goes up and down and up and down. Now we take the same, not the same string, but we take another string, all right, very long string, and we put one end here, and then we trace, all right, the microvilli all through this whole pathway here, all the way to this end. Then we cut this string, okay? And then when we stretch it out, it comes out to be 15 micrometers, all right? And it's only because it's going up and down and up and down and up and down.
Okay, so the cells just shaking themselves out.
Yep. Well, this is a apical modification. It's created this modification to the top surface, but it's shaped essentially over time. You know, if you believe in evolution, whatever, it's evolved into that.
It's like the pile of a run.
Yeah, exactly. I mean, we could, if you want to drive to Spartanburg and you want to get there fast, you take 85. But I could take the scenic route, take 29. You know, it's going to be more miles if I drive 29, but I'm still going to get to the same place. You know what I'm saying? This is the same thing, kind of. Yes, ma'am. This one here on the left is without the microvilli. Yep, I'm just trying to illustrate the usefulness of the microvilli. All right, so we saw non-ciliated. Now let's check out the ciliated. All right, now this is the cell that has, all right, the cilia on top, which are these hair-like projections that come off. Okay, and the purpose of the cilia is to move things. Now the cell is stationary; it's not going anywhere. Okay, but the cilia are going to beat back and forth. So anything that's outside the cell here, the cilia will move past the cell. Okay, so there's a reason for that. So we'll see if we have epithelium that has goblet cells, all right, and ciliated simple columnar epithelia has goblet cells. So that means it's going to make mucin, which will eventually become mucus. And so we got to get rid of that mucus. We can't have it sitting there. All right. You probably notice when you get a cold, you get that mucus build up. All right. We've got to move this mucus out of there. So the cilia will beat back and forth and move the mucus out of the way. So we'll see this in our bronchioles. All right. We learned a little bit about the respiratory tract. Right. The trachea. We learned about the primary bronchus or bronchi, I should say. We have your trachea, and then it branches off into a right primary bronchus and a left primary bronchus. Well, then those taper down into what we call secondary bronchi, and then we get tertiary bronchi, and then we get really, really small, tiny air passageways. Those are what we call bronchioles. Okay? So the bronchioles are lined with this type of epithelium. Okay? So we don't want mucus to get stuck down into our alveoli. All right? So this type of epithelium has the cilia, and it's going to get rid and move that mucus out away from the alveoli into your bronchi, into your trachea. You'll cough it up or whatever, either you'll spit it out or swallow it. Okay? But it doesn't want it to go down into the alveoli. You'll also see it in the uterine tubes. Okay? Remember we learned about back in chapter 1. Okay? The ovaries there are going to make the eggs, well, not make the eggs, but they have the eggs, and they're going to expel an egg into the uterine tube. Well, the egg is eventually going to make its way to the uterus. How does it do that? Bam, the cilia. The cilia are beating back and forth, and they're going to push the egg down into the uterus. All right? And so it takes about 6 days for that to happen. All right, so here you can see the little picture here. All right, this is an example here, all right, of our uterine tube right here. Here's the lumen. All right, you see it. I don't know if I can make that bigger. I can. All right, here you can see the cilia. It's not like a brush border. It's not nice and uniform. There's like divots and indentations. It looks rough. All right, well, that's how we know it's cilia. All right, and then the cilia will beat back and forth, and as the oocyte is traveling by it, the cilia will push it down and have it move further down the uterine tube, making its way to the uterus. Okay?
What's in the middle?
That is going to be some of our connective tissue, some blood vessels there. All right. Pseudostratified columnar epithelium. This is that type of epithelium that looks like it has multiple layers, but it is not stratified. Again, all the cells are in direct contact with that basement membrane. Here's the thing that makes it look, all right, stratified is because the nuclei are scattered at different distances. Some are high, some are low, some are high, you know, it's all over the place, okay? But this is the important thing. Not all of them will reach that apical surface, okay? It gives us the appearance that, all right, these are stratified, right, but they all don't make it to the apical surface here. All right, so the two types are ciliated and non-ciliated. So again, let's start here looking at our ciliated. So the ciliated are going to have cilia on the apical surface, and we know that cilia is present to move things. It is going to move things because the cells are going to be stationary. So the cells remain stationary, and the cilia will move things past the cell. All right, in this case, it's going to act for a protective functioning. So we'll see goblet cells because we have cilia there. So they're going to produce the mucin, which will turn into mucus. And we'll see this type of epithelium in our upper respiratory passageways. Nose, the nasal cavity, all right, part of your throat, down into your trachea. Okay, so the large passageways of the respiratory system. So here's a nice picture of it. Okay, so you can see now this at first glance when we look at this, look, you got nuclei here, up here, down there. It's all over the place. It looks stratified. That's why it can be very, very confusing. Okay, but again, all of these cells are connected to the basement membrane down here, but not all of them make it up to the apical surface. Now you can see here all the cilia, the hair-like structures here, right? And you can see we've got our goblet cells here. that are going to produce that mucin, which will turn into mucus, and the cilia will beat back and forth, all right, to move out of the way. Most of the time, it'll move it down into our throat, and you'll swallow it or spit it up. I know. Think about how much mucus you guys are probably digesting during cold and flu season.
So I had a mucus seal that I had to get removed.
Mm-hmm.
Is that those actual goblet cells? Like I watched them like pull them out.
That
It
one I'm
looks
not.
like it, honestly.
Yeah, that I'm not quite sure. I don't think so.
Okay.
I don't think it's going to be the specific cells because there's thousands, if not millions of them. Yeah, because they're so tiny.
Okay.
So goblet cells are found in both ciliated and non-ciliated.
Mm-hmm. Yep, and that's the thing, because when you see ciliated, think goblet cells. But the non-ciliated one, all right, again, because it serves as an absorptive function, how I always remembered it is it's found in the small intestine. And I just put it in my head, well, the goblet cells produce mucus, which helps to neutralize. It's not the main function of the mucus, but it helps to neutralize stomach acid. And that's just one of the ways that helped me to remember that it was found in the small intestine. All right. Let us go on to non-ciliated. This one's easy. It's non-ciliated, excuse me. So that means it has no cilia, no goblet cells. Okay? So none of that. So it also serves as a protective type of epithelium here. Okay? It's a rare type of epithelium. And you're only going to see it here in the the epididymis, which is that ridge structure on top of the male testes, and then we'll find it in the male urethra. Okay? So coming down here, that's what it looks like here. Now again, all right, it looks like it's stratified. Okay, but you have some nuclei up here, some down here, and it almost looks like in some areas they're just stacked on top of one another. But you don't see any cilia here, and you don't see any goblet cells. All right, so let's get into the stratified epithelium. We're almost done. Okay, so we have two types of stratified squamous epithelium. Your eyes are glazing. I know. Trust me. We're almost done. The harder stuff is out of the way. Like I said, I'm only going to do epithelium today, and then we'll start labeling here. All right. So, has anyone ever heard of the term crack kills? Seriously. Okay. I think most of us have. All right. Well, I'm going to teach you a new term. Keratin kills. Okay. And we'll learn more about that in chapter 6 when we do the skin. So, first of all, stratified squamous epithelium comes in two forms, keratinized and non-keratinized. So this type of epithelium, all right, that we're looking at here is the non-keratinized. Okay, so it does not have keratin. Keratin is a protein that specific skin cells will make to protect the cell tougher. Okay, but unfortunately, when it makes this protein, it kills itself because it fills up the inside of the cell with this protein. Well, there's no room for the nucleus of the cell, which is the control center, so it kills that off, and then it'll kill off some of the cell organelles. So when that happens, cell's dead, okay? We'll go into that in more detail, but just know that, okay? So that's why for this statement here, it says all the cells are alive, because it's non-keratinized. Those cells don't make keratin, so they don't kill the cell off. So you're going to find this type of epithelium, right, in moist areas. So your mouth, okay, your throat, okay, the anus, the vagina, areas where there's going to be moisture involved. Saliva, mucus areas here, okay? So normally, like I said, if a cell's living, you should be able to see the nucleus. Okay? Certain cells don't have a nucleus, red blood cells, for example, all right? They don't. But for our purposes, all right, if the cell has a nucleus, it's alive. If it doesn't have a nucleus, right now, we'll consider it dead, okay? I'll tell you when you need to consider it otherwise. So in these moist areas here, which are going to be under a lot of physical and mechanical stress, you're going to find it in your mouth, part of your throat, which is the pharynx, the esophagus there, the vagina, and the anus. All right? So I'm going to zoom in on this. So this picture here, all right, we're seeing it's a sample of the vagina, but you can see, look, all this is stratified squamous epithelium. Okay. Now these dark purple dots, those are the nucleus. Okay. So you see there's a bunch of nucleus down here. Then we go up to these upper layers here. You see some nuclei up here, but I don't see a nuclei here or a nuclei here. Are those cells dead? They're not. Now here's the thing, and I'm going to tell you why. Pretty messed up. Well, it's not messed up, but when I draw this picture for you, you'll understand why. All right? So here are some squamous cells here. Okay? These are supposed to be squamous. And I'll put a nucleus here, and a nucleus here, and a nucleus here, and a nucleus here. All right, now, I'm a histologist. And I want to make a slide. So I get my tissue, okay, and I'm going to make my slide. And so I'm going to take a slice down through, all right, down through. Yeah, you got these videos to watch, Alexia. You can watch these videos and you'll own it like a boss. All right, so I'm going to take my slide of that tissue, all right, with this thin slice, okay? And then you can see, did I get this nucleus? Did I get this nucleus in my slice? No, I didn't get this nucleus. I didn't get this nucleus. I got this nucleus. So just because it's not in that slice doesn't mean that that cell is dead. You guys kind of see where I'm going with now? All right, so when we go back down here, all right, we just, no pun intended, it didn't make the cut. The nucleus in these cells didn't make the cut. It was either to the left of the cut or the right of the cut or above the cut or below the cut. You see what I'm saying? All right, so this is what I'm saying. When you're looking, if you want to know that if this is keratinized tissue, look at the top layer. If you can see a few nuclei there, even one or two, all right, that means it's living, okay? You just have to assume that all these other layers below it are living. All right, I'll show you a picture of one that's keratinized. All right, let's go to the next one here. Boom. All right, here's our, oh wait, I didn't do that. Oh, wait a minute, that's non-keratinized, isn't it? I think in our lab book we have, in the atlas there, we might have a picture of the keratinized. Well, we'll see it in the skin. All right, so stratified cuboidal. Again, stratified, 2 or more layers, cuboidal, all right, square shape. I'm going to kind of skip that part here. So we're going to see, all right, this in tubes and coverings of structures. All right, mainly cuboidal tissue, think associated with glands, some sort of gland. Okay, so stratified cuboidal is going to be for protection and secretion. So it's going to make up the walls of ducts. Okay, because here's our gland here. All right, and then it's got a tube or a passageway. So we make all of whatever. If it's a sweat gland, it gets made by this part, and then it's got to send it out here. Okay? So this part is called the secretory portion, and this tube-like structure is called a duct. Not like a duck quack quack, but a duct, like duct tape. I used to think when I was younger it's called duct tape. No, it's not. Okay? Anyways, this type of epithelium forms the walls to these ducts. Okay? And so you'll see them in exocrine glands. An exocrine gland is a gland that has a duct. An endocrine gland does not have a duct. Okay? Your adrenal glands, adrenaline, that sit on top of your kidneys, those are endocrine glands. They don't have ducts. Okay? All right. Stratified columnar epithelium, rare type of epithelium. Okay? Again, 2 or more layers. All right? So these cells will appear to be columnar at the apical surface. They operate for protection and secretion. So again, think of this type of epithelium having to do with glands, but for the salivary glands, for saliva, the larger portion or duct part of that, or the duct part. All right, you'll find that also in the male urethra there. I think that's it. Oh, no. Last one, transitional epithelium. This is the transformer epithelium, okay? So I talked about it being in the urinary bladder. So we're going to see it in our urinary tract here. So it presents in two states, a relaxed state and a stretched state. So when it's empty, there's no urine there, okay, the basal cells, the cells at the bottom, are going to have a cuboidal or polyhedral shape, all right? And then the cells at the top are going to be large and rounded. And then when we fill our bladder up, they become flattened. This type of epithelium has a specific characteristic, all right, is that it has binucleated cells. And what that means is you'll have a cell with two nuclei in it, and we call it owl eyes. It'll look like an owl, something like that. Okay, so two nuclei that will be inside one cell. So this one, the one in your book is, I think, in your atlas is a little bit better than this one. But you can see, as we look at the transitional epithelium, right here you can see in an empty bladder, all right, the apical cells are more rounded, all right, they look fine. But then, I'm trying to think. Yeah, I can't really see a really good example of a binucleated cell here. That's why the one in your book's a little bit better. This one here is not bad. But when your bladder's full, those apical cells get squished. They're flattened. Here's a nice binucleated cell right there. Here's another one right there. All right. Here's another one right there. Okay. All right. I think that's it for boom. All right. So we're going to stop here and do some labeling. Let's give our brains a moment or 10 to relax here. And if you have your atlas at home, bust it out. Okay. And if you don't, you can't give me the excuse that you left it at home because you're at home. See what I'm saying? I'm on to them. All right. So let's do some labeling here. All right, so I'm going to tell you what I told my other class, that your lab class, the lab exams are 80% identification, okay? So a good majority of that material is, we'll put a slide up like this and we'll say, what is it? And you need to name it. So if you're kind of worried about the lab portion, I would say start off by being able to spell all these terms correctly. That's what the lab manual is for. It tells you how to spell it and really kind of go from there, okay? The other 20% is going to be true, false, multiple choice questions, you know? Perfect. Here's an example. Here's a lab identification. This next slide would say, where do you find this in the body? And that's where you would write in these responses here, okay? We'll come back to that slide in a second. So first off, here's our simple squamous epithelium, okay? Flattened cells. Okay. So these are nice flattened cells, flattened nuclei, right? So you're going to find this in areas where we're going to see absorption, filtration, and secretion. In the alveoli, the air sacs. Remember we talked about this before. The mesothelium. Oh, no. The endothelium, which is the lining to your vessel walls, blood vessel walls and the lymph vessel walls. And then also your serous membrane, okay, which is known as the mesothelium. Okay. So in the test, if you were to write in there endothelium, you'd be right. If you were to write in mesothelium, you'd be right. Just to let you know too, for lab tests, when you take your test and you get your score right away, that is not your final score. Listen to me at home, that will not be your final score, so don't freak out. Okay? Because if you get like a 20 on it, don't worry about it, unless you know that you just haven't studied, and then you should worry about it. All right? But it's because the computer is very specific, all right, on certain things. Like, perfect example, when you do the muscles, after every muscle name, you have to put M dot. But if you were to write out the rectus abdominis and then the word muscle, it would mark you wrong. Okay? It wants you to write M dot. So anyways, I go through and grade them individually. So that's the point. don't freak out if you get a low grade in the beginning. All right, everybody always winds up with a higher grade once I go through and grade. But it takes me a couple days to go through it. So it may take you about 4 or 5 days to get your test results back. Okay?
So we'll like never have tests or quizzes again.
No, you'll take all your stuff at home. Have you taken the honor lock practice test? If you guys haven't, take it. All right? It's not a graded thing. It's just a practice test, so you know how to use honor lock. All right, that's going to be the proctoring program that you're using. You don't have to download anything either. All right, you don't download anything on your computer for honor lock. That's one of the reasons why we use it. All right, the next type of epithelium here is our simple cuboidal. This is a great example of simple cuboidal epithelium. All right, look at these cells. They're wonderful to look at. All right, they're as tall as they are wide. You get a nice spherical nucleus at the center. This dark black line here that you see, that is your basement membrane. And this space here, that's the lumen, right, of whatever this tissue is from. Okay? All right, so where do we find this? All right, line into the thyroid gland, kidney tubules, because our kidneys are hugely responsible for filtering things out. Looks like an octopus leg.
Are the exams going to be like actual slides, like these photos,
Yep.
or the?
It's going to be, I can't guarantee that there won't be some from the book, but it'll mainly be your lab atlas slides. There might be a few, all right, a few. And when I say a few, maybe 3 or 4. And I stopped saying none because sometimes, because these are randomized tests, all of you are going to be taking, you won't have the same exact test. It selects the questions at random from a pool. So you might get different ones than this person over here or this person over there, okay? All right, so that's our simple cuboidal epithelium, right? You're gonna see it, just think of glands here, all right? All right, here's our non-ciliated simple columnar epithelium. Great example. You can see only one row, all right, of epithelium, okay? Columnar in appearance because they're taller than they are wide. The nucleus is the basilar region. Here's a goblet cell here. You can see its nucleus is way down here. Here's another goblet cell. And then here, you can see the microvilli. There's the brush border right there. All right, brush border. Okay, so you're going to find this type of epithelium pretty much in the digestive tract from the stomach down to the anus. This dark brown line here, this structure, that's your basement membrane. Right, so here you can see, here's an example of a goblet cell. And then this is indicating the microvilli. Now, at the same time, this is indicating the cilia. And I'm not a huge fan of that.
Right.
Yeah, I know. This is supposed to be non-ciliated columnar. So, again, I can't say that this might not show up on the test. When it's me making the tests, when pre-COVID, I don't show this slide. Okay? But if you see this appear on the test, all right, and it's pointing to these structures here, all right, it's not cilia. All right, I'm just telling you, this is, I'm just saying, for this, pardon?
That would be the answer.
But yeah, that's what I'm saying, because this might be in the test pool. That's why I show it. All right, but this is not cilia. It's part of when they were making the slide preparation. That's just some damaged tissue that was in inappropriate slide preparation.
So are we calling it cilia or not?
You're going to call it silly if it shows up on your test. All right, and again, I don't show it normally. They lie to us. Yes, I know.
But this is an example of non-ciliated symbol.
Yep. So yeah, this epithelium is non-ciliated. That's what I'm saying. I don't in my test, like when we used to do tests, it always does that. But here's the thing. if you get this line on your test, just whatever this is pointing to, that is cilia, okay? But it's not. But it's labeled on this because, again, there are certain things that I just want to make sure that you guys see in case some of the things slip through the cracks there. All right, there's your basement membrane right there, indicated by that dotted line there. All right, here is the non-keratinized stratified squamous epithelium. Remember, non-keratinized. This is all living. So again, look here. All right, this is the lower portion of the epithelium, the basilar region. And then as we go up, you can see, all right, here's some nuclei, but there's a whole bunch missing here. Remember, those didn't make the cut. But as we get to the top portion, you can see there's a nuclei here. Here's one. Here's one. Here's one off to the side. Point being is, all right, this is non-keratinized. All right, even though there's a bunch missing, all right, in some of these layers, it's only because it was to the left or the right of the slice. Okay, always look to the top to see. Yes, sir.
Is there a reason why in the book it doesn't say non?
Again, yeah, because to be honest with you, usually when we discuss keratinized and non-keratinized, it's in chapter 6 when we go through. All right. But technically, it should say that just to show you, because technically it is, you know, a type of stratified squamous epithelium, specifically the non-keratinized type.
Is there a visible basement membrane?
Nope, you can't really see it. All right, so you're going to again see this in moist areas. So your oral cavity or the mouth, all right, the pharynx, throat, part of the esophagus there, all right, the anus and the vagina, okay? You're going to see it in areas where mechanical stress can be pretty severe. And what I mean, your mouth takes a beating. I mean, you think when you eat Jell-O, not so much, but when you're eating crackers and popcorn, you're tearing all these outer layers here, you're sloughing it off, all right, and you're beating it up. All right, here's transitional epithelium. This is an example of an empty bladder. You can see the apical surface, all right, is nice and rounded. It's not flattened, but there's some great examples. Here's a great example of a binucleated cell right here. You can see the owl eyes here, all right. This one's okay. Every time, oh, here's the one. Here's a nice binucleated one. Here's another binucleated one. Another binucleated one. All right. So I like this one better than the one in the book. Should you write that in? All right, in the urinary bladder right here. Okay, that's where you're going to find this type of epithelium. All right, here's an example of the pseudostratified ciliated columnar epithelium. That's a mouthful here. But if you look here, okay, a lot of, and this is not the best slide, and I very rarely use this slide, but you can see, all right, the nuclei are, some are high, some are low, all right? They're just stacked in different positions, okay? So remember, with pseudostratified, all of these cells that you're seeing here are connected to the basement membrane down here, okay? But not all of them are making it up to the apical surface here. Again, it's tough to see, and if I zoom in on this, and I've tried, the resolution's not very good, but this is all cilia up here. So this is not a very high magnified slide here. But that is cilia up there. Okay, you can see some goblet cells here, 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 here. All right, so you're going to go and see this type of epithelium, all right, in the larger passageways of your respiratory system. So your nasal cavity, all right, part of the throat there, all right, your larynx, which is your voice box, trachea, some of the bronchi there. We don't have a picture of it here, but the non-ciliated type, that's more rare, okay? That's going to be epididymis and part of the male urethra, okay? We just don't have a picture of it here in the book. All right, glandular epithelium. If you look at this epithelium, what else does it resemble? What's it look like to you? What does this look like? Let me zoom down.
It's like one of the cell shape.
Yeah, what do you think? Give me a cell shape. Would you say, well, let's do this. Simple or stratified? Okay, I'll go with simple. Simple, yeah. Yeah, okay, so it is simple, but this is cuboidal. Let me just zoom back out. So in this case, and again, for me, and I tell folks, unless I'm specifically asking you for the epithelium, all right, and I would word it in a way that pretty much you'd have to write down glandular, all right? If you were to write down glandular or simple cuboidal, you'll get credit with me, okay? So you're going to find this, all right, in some sort of gland, thyroid gland, perfect example. Because this is actually from the thyroid gland, and this is thyroxine that's being produced right here in the center. Okay, so again, this type of, so if you could write down glandular epithelium if you know it's glandular tissue, but there's no way of you knowing that unless we put it in the question. All right, this tissue was taken from the thyroid gland. Then you would say, oh, okay, then it's glandular epithelium instead of simple cuboidal. Okay.
So when you have like hypo and hyperthyroid issues, is that something that the tissue controls, or is it something like is it resulted?
Yeah, and it could be both. All right, because there could be something wrong with the gland itself, that it's not producing enough thyroxine, if we're talking about the thyroid, but it also could be something wrong with the hypothalamus, which is the master gland, all right, which controls almost all of the endocrine functions in your body, and then the pituitary gland, which is part of that. Going back to the negative feedback loop, okay, it's part of that, and you're going to learn in much more detail in chapter 17, when we do the endocrine chapter, you're going to learn that negative feedback loop and what could cause hyperthyroidism or hypothyroid. Because if it's not producing enough thyroxine, it could be a problem with the thyroid gland itself. Or maybe it's the pituitary gland or the hypothalamus, which is not telling the thyroid gland to make thyroxine. So there's a couple different possibilities. But yeah, one of the possibilities could be the gland's just not working. All right, stratified cuboidal epithelium. Okay, you can see now this isn't the best. It's not the worst, but it's not the best. All right, these epithelial cells are going to be cuboidal in shape. All right, because you can see there's nice spherical, and some of these examples, that's a nice spherical oval nucleus. Excuse me. That's a nice spherical round nucleus. This one's kind of round. All right, so there's not a lot of space between this layer here and this layer here. Because remember, stratified or cuboidal epithelial cells are as tall as they are wide, where columnar are taller than they are wide. So there's going to be a lot of space between the nuclei. And I'll show you. Look at this here. Let me come back to this one in a second. And look at this one here. Here's stratified columnar. You can see how much space there is between the two rows of nuclei. Here's a nice row here. Here's a nice row here. All right, because of the shape of that epithelium, we'll have some space in between. But you can see, well, it doesn't matter. I can go back to this one here. All right, there's not much room between because these are much shorter cells. All right, so again, this is a duct wall here. So this is part of a gland here. Just think that this type of epithelium is going to be found around exocrine glands. Okay. All right. I think this is the last one. Nonciliated, stratified columnar epithelium. All right, you can see, all right, we have two rows here. Okay, this stuff is pretty rare. Okay, and again, you're going to find this with glands, the larger ducts of salivary glands, okay, like the parotid gland. All right, again, parts of the male urethra. Pretty rare, pretty rare. Yep, all right, that's it for epithelium. That's it for epithelium, everybody at home. It's a lot. Epithelial and connective tissue can be somewhat frustrating because there's a lot, but I'm telling you, once we get to muscle and nervous tissue, those are much easier. So it won't be as bad, trust me. It goes from being difficult to a little bit easier. Yeah, I know. We're gonna have some classes like that. It's gonna be like a marathon. Any questions about anything? I'm gonna stop the recording here.